Hello students, welcome to EPG Pathshala. I am Dr. Zeenat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to discuss about the module Introduction to Transdermal Drug Delivery System from the paper NDDS Part 1. In this module, we will talk about number 1, the transdermal drug delivery. We'll try to understand the various aspects of transdermal drug delivery. Then we will try to understand the basis of the skin. Skin is primarily the site of the transdermal drug delivery. We'll try to understand that what is the anatomy and the biochemical aspects of the skin. Later on, we would focus on the various mechanisms which are used for the drug permeation through the skin. Towards the end of the module, we will highlight about the factors of the transdermal bioavailability. There are a gamut of factors which are responsible for the availability of the drug through the transdermal route. Therefore, we'll try to understand each one of these factors one by one. We will also highlight about what are the various advantages, what are the limitations of the transdermal delivery, and we will go in for highlighting the various pharmaceutical applications of transdermal delivery. The most extensive route of drug delivery is the oral route. It has a lot of advantages, most important being the patient compliance. This route has, however, got a number of disadvantages, including enzymatic, acidic, and basic drug degradation in the GIT, first-pass metabolism, incompatibility in uncautious patients, and the patients using polytherapy. The difficulties faced by oral drug delivery system could be overcome by a novel drug delivery system. In recent years, the use of intact skin has been explored as a potential route of drug administration, and the drug delivery into the systemic circulation in a very continuous and a controlled manner is a successful option. The transdermal drug delivery system can be defined as a system which, when applied over the skin, delivers the medications across the skin in a controlled and a predetermined rate in order to minimize the adverse effects and maximize the efficacy of drugs. US FDA has long approved the first transdermal patch, which comes by the name of Transderm Scop, which contains scopolamine for the treatment of motion sickness in 1979. Transdermal drug delivery system formulations aim to maintain the drug concentration within the therapeutic range in the systemic circulation because of the continuous and the controlled delivery. The skin is the primary site of the transdermal delivery. We all know that skin has a huge area of around 2 meters square and has an average thickness of about 2.5 mm. It constitutes 6% of the total body weight, which amounts about 5 to 6 kgs. The skin pH is somewhat acidic, falling in the range of 4.2 to 5.6. It provides barrier against a range of noxious stresses like the UV radiation, the mechanical, the chemical, and the biological insults. It acts as the periphery sensing system. It maintains the body homeostasis and acts as a barrier to prevent the desiccation and helps in maintaining the temperature balance and offers protection from the UV radiation, while absorbing the pigmentation system complex. It offers immunoregulatory network protection as well. It is preferably a beneficial route to deliver drugs into the blood circulation with the least proteolytic activity. The transdermal drug delivery is usually hampered mainly by the barrier of the stratum corneum. This is the dead skin layer, the topmost one. The flexibility on the other hand of the skin promotes the permeation of the drug across it without permanent distortion, and its thinness is enough to cause stimulation. The screen in front of you depicts a very typical and a classical diagram of the skin, where probably you are able to appreciate the two primary layers which are primarily present in the mammalian skin, namely the epidermis and the dermis. Towards the end, you are having the subcutaneous tissue. It also is highlighting the presence of the hair shaft, showing the sweat pore, the papilla of the hair, the blood and the lymph vessels, and finally the sweat glands. Let us now dwell a little deeply into the topmost layer of the skin, and which is referred to as the epidermis. It primarily consists of number 1, stratum corneum, which basically comprises of around 15 to 30 sheet of a non-viable but biochemically active corneocytes. Following this is stratum granulosum, which comprises of 3 to 5 sheet of non-dividing keratinocytes and produce keratinohyaline. The third after this is referred to as the stratum spinosum, which is primarily consisting of 8 to 10 sheet of keratinocytes and which has got a very limited dividing capacity and is also a seat for Langerhans cells. The last of this is referred to as the stratum basale, which is a hub for maturing and aging keratinocytes, melanocytes, the Merkel cells, which are also referred as the receptor cells. To sum up, the epidermis is an external, stratified, non-vascularized epithelium with a thickness ranging between 75 to 150 micrometers, and this undergoes a continuous process of keratinization. Just a while ago, we just studied about the epidermal layer. This epidermal is probably the first permeability barrier for the final functioning of the transdermal drug delivery. The human epidermis offers a permeability barrier which depends upon transglutaminase mediated cross-linking of the structural proteins and lipids. These are also referred to as the biological glues. The transglutaminase mediated bonds exhibit a high resistance towards the proteolytic degradation. Proteins are then highly resistant to mechanical perturbation and proteolysis. The quality of the stratum corneum barrier, the stratum corneum, that is the topmost layer, depends upon the presence of equimolar concentrations of ceramides, cholesterol, and fatty acids. We must understand that the changes in the concentration of any of these can affect the barrier quality. So whenever we talk about the transdermal drug delivery, we need to understand the barrier which is offered by the epidermis of a normal skin. Then we can devise means and ways that how we can surpass or circumvent this barrier. Following the epidermis is the dermis layer, which has got a major role to provide the much needed tensile strength to the skin. It primarily regulates the temperature and supplies the epidermis with the nutrient. The body's water supply mainly is stored within this dermis. It has got the major components like the hair follicle, connective tissue, sweat glands, sebaceous or oil glands, apocrine glands, lymph vessels, and the blood vessels. The primarily cell type in the dermis is referred to as the fibroblast. The dermis protein primarily constitutes of the collagen protein, which is a total of 90% of the total dermal proteins. It also has elastin, proteoglycans, glycoproteins, water, and hyaluronic acid, which primarily comprises the extracellular matrix. The skin appendages. After understanding the presence of the epidermis and dermis, let us go into the understanding of the skin appendages. We all know that skin plays an important role in the body homeostasis. Therefore, it is very well equipped with the secretory and excretory capacity. The secretory capacity primarily includes the release of chemicals from cells for the physiological functions, while the excretory capacity comprises the elimination of waste products of metabolism. The sweat glands usually secrete the sweat, which has got a strong odor. If it is having a strong odor, the secretions are called as apocrinic secretions, while something which is a faint odor, it is referred to as the acrinic secretions. The sebaceous glands secrete sebum onto the hair follicle to oil the hair. And finally, the other skin appendages is referred to as the hair follicle. Let us now try to get into the understanding of the sweat glands. You will be surprised to know that a human skin consists of around 3 to 4 million of acrine sweat glands, and each producing a water perspiration, which usually help us to cool or keep our cool. It maintains the core temperature at the desirable 37.5 degrees centigrades. The diagram in front of your screens primarily gives a highlight of the presence of the sweat glands in the dermis area of the skin. At maximum, the output of the acrine sweat glands can be as much as 3 liters per hour, and the heat loss is more than 18 kilocalories per minute. The humans utilize acrine sweat glands as a primary form of cooling. The apocrine sweat glands are larger, they have a different mechanism of secretion and are limited to axilla and the perianal area. The composition of the sweat primarily consists of the macro elements like sodium, potassium, calcium, magnesium, whereas it also has little amount of micro elements including zinc, copper, iron, chromium, nickel and lead. The sebaceous glands. You can see that they are present in very close proximity to the hair follicle. The glands are usually secreting an oily or a waxy matter which is referred to as sebum, which does a job of lubricating the skin and the hair surface. The composition is usually consisting of wax, monoesters, triglycerides, free fatty acids, squalene, and a very small amount of cholesterol esters and cholesterol. Following the dermis tissue lies a layer of subcutaneous tissue which could be understood as a sheet of fat containing the areolar tissue known as the superficial fascia, and it attaches the dermis to the underlying structure. Together, the epidermis, the dermis, and subcutaneous tissue forms the human skin. The skin is probably also involved in metabolism. So we have a process which is referred to as skin metabolism. The primary source for energy production in the epidermis is normally glucose, which is obtained from the circulation. The keratinocytes take this glucose, or the glucose diffuses into the keratinocytes without the effect of the insulin. The large proportions of glucose is catabolized up lactate even in the presence of oxygen. The secondary source of energy in case of skin metabolism is derived from the fatty acids which are stored in the epidermal tissue or sometimes from the exogenous sources. This happens only when the glucose flow is limited, and then only the fatty acids are metabolized. The immune system in the skin. It is but understood that the skin also has an immune protection. It not only provides the immunity for itself, but also for the whole body. The cell types which are present on the skin contain battery of mediators of the immune responses. The cells which are primarily involved in the immunity are the Langerhans cells, monocytes, macrophages, and mast cells which work in cooperation with the T cells. The cell types producing the free radicals, antibacterial peptides, cytokine chemokines, pro and anti-inflammatory mediators include the neutrophils, eosinophils, and the basophils, while the B cells secrete the immunoglobulins or the antibodies. My dear students, as we have already decided that we have to understand about the transdermal drug delivery, before we get into the details of the drug delivery, how it happens, let us try to find out the various mechanisms which are involved in the drug permeation via the skin. Skin being the site of action, it is very important for all of us to understand that how it pretends or how it proposes itself while a drug carrying drug delivery system is attached to it. The skin physiology primarily suggests three possible pathways by which the drug is passively diffused across the skin and deep into the blood circulation. The diagram in front of you proposes these three mechanisms. The first one, as obvious in the diagram, is the intercellular diffusion through the lipid layer, followed by something which is called as your transcellular diffusion through lipid bilayer and keratinocytes. And the third mechanism proposed is diffusion through the hair follicles and the sweat ducts. The drug traverses through any of these mechanisms depending on its inherent behavior. The lipophilic drugs pass mainly through the lipid matrix present in the stratum corneum, and while the hydrophilic drugs permeate through the hydrated stratum corneum by the polar pathways. In continuation of what we have just understood, let us try to predict that how a transdermal patch, which is the commonest type of the transdermal drug delivery system, will probably go through the process of transdermal permeation. Once the patch is attached or a transdermal drug delivery system is attached, which primarily is a composite system, the drug from there would transverse through or traverse through the stratum corneum and the epidermis, followed by its path into the dermis, and as it reaches down, it is from therein distributed to the various sites, which can be designated as the depot site, the site of action, into the blood capillaries, the metabolite site, and finally the elimination. Just a few minutes ago, we have been able to understand that what are the three basic mechanisms for the delivery of the drug into the skin. There are a host of factors which actually affect the final availability of the drug or the transdermal bioavailability. There are a group of factors which play a very pivotal and important role, and these factors are summed up as the physiological factors, the formulation factors, and the physicochemical factors of the drug. We will try to group them separately and then try to understand each one of them. The first group of factors are referred to as the physiological factors, which involve a gamut of things like the hydration of skin, the skin temperature, the age of the skin, the blood flow, the regional skin sites, the level of skin metabolism, the differences of species, the condition and pathology of the skin, and finally, the flora and enzymes of the skin. Amongst the physiological factors, the first factor is the hydration of skin. Hydration of the skin is important in causing the swelling of stratum corneum of the skin. It also provides fluidity to the skin. It also increases the solubility and partitioning from the vehicle to the membrane, and this drastically helps in permeation of the drug molecules through the skin. The next factor is the skin temperature. If the temperature of the skin gets increased, the percutaneous absorption of the drug also increases due to the vasodilation of the blood vessels and the fluidization of lipids which are in contact with the skin. The increase in the blood flow to the skin increases the absorption of the drug through the skin. The next factor is the skin age or age of the skin. The young and the elderly skin shows more permeability than the middle-aged persons. Infants and children are more sensitive to toxic effects of drug through the skin because of the absence of stratum corneum in them. Blood flow. An increase in the blood flow helps in increasing the concentration gradient of the drug across the skin, by which the total time of residence of the drug in the dermis reduces by continuously removing it from therein. The next factor is regional skin sites and the species differences. The skin differs in anatomical features such as thickness and nature of stratum corneum, number of sweat glands, and number of hair follicles per unit surface area. This difference exists from person to person, species to species, and even site to site of the same person. This is the reason that in all cases, percutaneous absorption of drugs differs from one another. Keratinization of skin also varies from species to species. The skin metabolism and also the skin flora and enzymes. Hormones, steroids, chemical carcinogens, and some drugs are highly metabolized in the skin. Skin metabolism is important to determine efficacy of the drug permeated through the skin. There are various metabolizing enzymes and microbes which are present in the skin that metabolizes the drugs passing across the skin. There are only a few drug candidates that reach inactive form in the circulation. Otherwise, drugs are mostly metabolized to various extents in the skin. For an example, 95% of the testosterone gets metabolized in the skin before absorption. The next set of factors which are playing an important role in our overall transdermal drug bioavailability are referred to as the formulation factors. The primary amongst them is the release rate of the drug. The affinity of the drug with the carrier determines the rate of release of drug from the formulation. The physicochemical properties of drug like solubility in the solvent and interfacial partitioning from the formulation to skin determines the rate of release of the drug. Ingredients of the formulation is the next important factor. The formulation primarily consists of the polymers and the various set of excipients. These are present in the formulation and which would finally decide or affect the drug release or the drug permeation through the skin by modifying the physicochemical properties of the drug or altering the skin physiology. The third factor pertaining to the formulation is the presence of permeation enhancers. The permeation of drug through the skin is increased by using permeation enhancers, which could be of different categories. Permeation enhancers act by altering the integrity of the skin, that is, the physiological and physicochemical modification, temporarily to open the skin pores for drug absorption. Permeation enhancers may be chemical substances that act chemically or physical permeation enhancers which interact physically with the skin integrity. The next set of factors are referred to as the physicochemical factors of the drug. The primary amongst them is the pH, diffusion coefficient, partition coefficient, drug concentration, molecular size, and shape. Beginning with the pH, the dissociation of weak bases and weak acids is determined by the pH and pKa or pKb values. The proportion of unionized drug basically determines the drug concentration in skin. Diffusion coefficient at a constant pH and temperature depends on the properties of drug, diffusion medium, and their interaction. Partition coefficient. The permeation of drug across skin is directly proportional to its partition coefficient to a certain extent. An optimum partition coefficient is important for a drug to permeate through the skin. Drug with high partition coefficient K will not leave the pores of lipids in the skin. Drugs with low partition coefficient K value will not be permeated through the skin. The next factor in line is the drug concentration. The flux is proportional to the concentration gradient across the barrier, and concentration gradient will be higher if the concentration of drug will be more across the barrier. The drug absorption through the skin occurs by passive diffusion. Drug moves across the skin through the concentration gradient, that is from high to low concentration, so that the drug concentration in the formulation applied over the skin determines the rate of diffusion across the skin. Therefore, more the concentration of the drug, more will be the permeation through the skin. Molecular size and shape. Higher the molecular weight, lesser is the drug absorption. Similarly, small molecules penetrate faster than the larger ones. The table in front of your screen sums up the various ideal properties which will help us to have an excellent transdermal delivery. The transdermal drug delivery system has got very significant advantages associated with it. Transdermal drug delivery is a superior way to deliver drugs that are degraded down by the stomach acids and not well absorbed from the gut, or extensively first pass metabolized by the liver. So, what we can conclude, the drugs which are not suitably given by the oral route can precisely be given through the transdermal drug delivery. The transdermal delivery systems can be used to deliver drugs in the patients with constipation, dysphagia, or who are unconscious. So, patients who have got peculiar problems as just mentioned will be very suitable candidates for the usage of transdermal systems of drug delivery. Topical patches, which is one of the most commonest form of the transdermal delivery systems, are non-invasive and painless way of drug delivery into the body. The transdermal delivery could be a useful way of delivering the drugs in the patients who are polymedicated. So they have some drugs through the oral route and some drugs probably can be given depending upon the instructions of the physician through the transdermal route as well. Transdermal delivery system release medications in a controlled and steady manner over a prolonged period of time. The commonest form of the drug delivery in transdermal route are the transdermal patches, which could be cost effective. The transdermal patches are productive, especially when the course of treatment is long term, and such in cases of hormone replacement, chronic pain, treatment and smoking cessation therapy, etc. There could be an easy termination of drug therapy at any point of time, simply by removing the transdermal patch, especially when the toxicity is observed. This becomes an important attracting for the patient who uses them. A large surface area could be provided by using a transdermal patch as delivery tool in order to improve its bioavailability. The designing of a transdermal drug delivery system is having a multifaceted approach. So depending on our need, we can have a shape and a size as per the requirement of that particular drug. The topical patches additionally offer an advantage with fewer side effects compared to the oral medication or supplements. Of course, it has got a flip side. The limitations of the transdermal delivery should also be understood. The transdermal patches cannot be used as means of delivering ionic drugs because of the possible irritation and allergic reactions. So, the limitation is that if a drug is causing a possible irritation and allergic reactions, so it is not a good candidate for transdermal delivery. The transdermal patch system cannot achieve high blood plasma concentrations. So if there is a requirement that the drug has to be in a very large concentration, it becomes absolutely difficult to deliver such an amount through the transdermal patch. So that precisely becomes a limitation. The system cannot be used to deliver drugs of large molecular size. So we have another limitation, that it is suited for drugs which are precisely having a small amount of molecular weight. If it crosses that size, then precisely the system has got its own limitation. pulsatile drug delivery fashion cannot be accommodated or achieved by using the transdermal patches. I'll just try to emphasize here that if I want a drug to be delivered in a pulsatile manner, then transdermal patch is not the choice of the delivery system. It would precisely give the drug and maintain it in a controlled fashion over a prolonged period with not much change in between. The transdermal patches, on the other hand, can cause hypersensitivity reactions. It has to be put on the skin. Many of us are very, very hypersensitive. Therefore, this could be a very important limitation. There can be allergy, local irritation at the site of administration, such as itching, erythema, which is like redness, local edema, which is collection of the water in the local site when exposed to drug or the excipients present in the formulation. If we continue to talk about the limitations, another major limitation is the skin barrier activity. We understand that the skin barrier activity changes from gender to gender, from person to person, from the same gender at a different age group, and even from one side of the same individual. It simply means that the skin of a person X cannot be similar to the person Y. Not only that, the different sides of the skin of the same individual also have got different types of permeability. So this becomes a limitation for the transdermal drug delivery. If it is to be delivered at site A, it might deliver in an X manner. If it is to be delivered from site B, the change in the delivery approach absolutely becomes different. The poor skin permeability also limits the number and choice of the drugs delivered by the transdermal route. Skin permeability, whatever we do and whatever we say, still remains a limitation. Transdermal drug delivery system is somehow restricted to potent medicaments. So what we can say here is that the transdermal drug delivery system is not suitable or amenable to all types of medicaments. The transdermal drug delivery also requires a significant lag time, which could be an important limitation. Historically speaking, the transdermal drug delivery has long back established itself as one of the most premier novel drug delivery systems. The commonest form of the transdermal delivery happens through a transdermal patch, and there has been a large number of products which has been in the market for quite a long time. Amongst them, the nicotine transdermal patch is one of the highest selling transdermal drug delivery patches in the United States. This was made for releasing nicotine in a controlled manner for the use in cessation of the tobacco smoking. Then, another product which consists of an opioid medication like fentanyl comes by the name of Duragesic. Then you have another drug named by buprenorphine, which comes by the name of Butrans, are used to provide relief for severe pain around the clock. Similarly, estrogen patches have been prescribed to treat menopausal symptoms and postmenopausal osteoporosis as well. The nitroglycerin patches were long prescribed to treat angina instead of the sublingual pills. The non-steroidal anti-inflammatory drug ketoprofen and antihypertensive drug clonidine are available in the market in the form of transdermal patches. There are many categories of the drugs which have been successfully formulated into the transdermal drug delivery systems. Amongst them is the selegiline, which is a monoamine oxidase inhibitor and is probably the first antidepressant drug which has been formulated in a transdermal drug delivery system. Similarly, the hormone delivery of the market formulations in the form of the name Ortho-Evra or Evra is also available in market in the form of transdermal patches. This system has also been exploited for the treatment of the disease attention deficit hyperactivity disorder. The market of transdermal drug delivery system is significantly in upward trend that and will probably continue for the foreseeable future because of the potentially satisfactory patient compliance. There is a continued increase in number of transdermal drug delivery products which will continue to deliver real therapeutic benefit to patients all over the world. There are more than 35 transdermal drug delivery products approved in the United States for sale and nearly 16 active ingredients approved for use in transdermal drug delivery globally. Students, let us now try to understand that transdermal drug delivery systems are indicated for certain specific conditions. They cannot replace delivery completely, but they have found popularity because of the sheer patient compliance. Let us try to understand the various conditions where this system is indicated. The first category where the patient usually can go for transdermal drug delivery is something when the patient has intolerable side effects because of the oral medications, which might include constipation. It can be very well used when the patient is unable to take oral medication and maybe suffers from dysphagia or vomiting. It is indicated when a patient actually needs an alternative method of drug delivery because of some very cogent specific reasons. It is very nicely used for pain control, which is usually improved because of its reliable administration through this mode. The system might be useful in patients with cognitive impairment or those who for other reasons are not able to self-medicate with their analgesia. It is used when a patient is having chronic illness and requires chronic therapies for a long duration. For those patients, transdermal could be the best approach to deliver sustained and controlled release of formulations. It can also be very successfully used in combination with other enhancement strategies to produce synergistic effects. Usually, this is absolutely expected to be used when the route of administration is invasive, like intramuscular or intravenous injection, and this can very well replace these invasive routes of administration. It is used when the medication for treatment has got a very low oral bioavailability and good skin penetration properties. The drugs, even if made into the oral formulations, do not serve its purpose of being a good pharmacodynamic agent. Therefore, because of its enhanced or appreciable skin penetration properties, such drugs are preferably formulated into a transdermal drug delivery system. We must appreciate that it is not that transdermal patches or transdermal drug delivery systems are used for all the conditions. There are certain situations where these systems should not be used. What are these conditions? The first condition is when there is an acute pain. Because of its inherent lag phase of drug delivery, it would not be suitable if a condition is of an acute pain. It should not be appreciated when short treatment course is needed, therefore is more suitable for chronic therapy. Of course, it is not a reliable method when the treatment requires rapid dose titrations. It should not be used when the requirement of dose is less than equal to 30 milligrams in 24 hours. It is also not indicated when the drug for the treatment has got good oral bioavailability. The table in front of you sums up the various categories of the drugs and the indication of diseases for which the successful use of the transdermal drug delivery system has been made. You can see that the drugs like simple anti-inflammatory, diclofenac diethylamine, has been formulated into a transdermal drug delivery system, and also this system has been found very useful for diseases like Parkinson's, major depressive disease, motion sickness, etc. My dear students, let us now summarize what we have learned from this module. The first and the foremost thing which we have understood is that what is transdermal drug delivery. We have spent enough time on finding out the various nuances and aspects of the skin anatomy and its biochemistry. Of course, we did not deal about biochemistry in detail, but we could find out and understand the anatomical format of the human skin. We particularly also emphasized upon the barrier properties of the skin. The skin, probably where the first transdermal system is attached, is the first primary barrier. So we also try to understand that despite this barrier, what are the mechanisms of the transdermal drug delivery through the skin? These were thoroughly discussed. The various factors which affect the transdermal drug bioavailability were also discussed. There are a gamut of factors which probably have to be considered while going in for a transdermal formulation. The physicochemical properties of the drug would be there, the physiological properties of the skin are to be taken care of, and also the formulation aspects are to be considered thoroughly. We also suggested the ideal properties of the drug which would make a very suitable candidate for the transdermal drug delivery. Later on, towards the end, we studied in detail the important advantages and limitations of the transdermal delivery. We were able to thoroughly appreciate the successful commercial application of the transdermal drug delivery. We could understand that there are a variety of diseases for which this system is being found suitable. To finally conclude, we can say that a transdermal drug delivery is an important and integral part of novel drug delivery system. Rather, I would say that this is one of the most established component of the NDDS, and it is described as a self-contained discrete dosage form which, when applied to the intact skin, will deliver the drug while it goes through the skin into the systemic circulation at a very controlled rate. This type of drug delivery is highly required and appreciated in a variety of diseases. So this system can become very helpful in treatment of diseases which require chronic therapy. Thank you.